Welcome to Translational Conversations from Model to Medicine, a series of discussions between animal and clinical researchers on the important interplay of their approaches for making scientific advances in biomedical research. Through this series, we highlight how both animal and human subjects are essential to understanding the origins of illness and the development of novel, effective treatments, while we explore also how researchers from both sides can collaborate and support one another. I'm your host, Dr. Rachel Perleski, a neuroscientist and the 2025-2026 Brad Fellow. Today, we are joined by Dr. Lana Ruvolo Grasser Winters, an assistant professor in the Department of Psychology and the Ben L. Silberstein Institute for Brain Health at Wayne State University, and Dr. Millie Rincon-Cortes, an assistant professor in the Department of Neuroscience within the School of Behavioral and Brain Sciences at the University of Texas, Dallas. Dr. Ruvolo Grasser is a researcher investigating the biomarkers of risk and resilience for trauma-related psychopathology and the development of relevant treatments in human youth. Dr. Rincon-Cortes is an animal researcher leveraging rat models to investigate normative and adversity-induced plasticity of maternal behavior and reward neural circuitry in the rat dam and downstream effects in the developing offspring. Thanks to you both for joining me today. I'd like to begin by hearing you each describe in your own words what the major goals and objectives of your research are. Dr. Rincon-Cortes, why don't we begin with you?
Sure. So our big picture question that we're interested in understanding is how adversity exposure during sensitive periods across the lifespan program maladaptive behaviors. So we focus on the postpartum period because it compromises caregiving, and we know from clinical literature that compromised caregiving has the potential to initiate pathways to psychopathology in the offspring. So we're interested in not just what behavioral changes occur as a result of adversity exposure, but being able to discover what the neural changes are that are driving these behavioral effects with the goal of developing novel interventions to try to prevent those adverse effects.
Fantastic. Thank you. And Dr. Grasser, why don't you tell us about what the major goals and objectives of your research are?
Sure. So we're really interested in similarly how those adverse life experiences impact the developing nervous system of children and adolescents, trying to figure out what might change in the structure and function of our nervous system, allowing us to perceive and respond to the world around us in different ways, how we can leverage information about those changes to know who might be at risk for something like post-traumatic stress or anxiety disorders, and then teaching us about what interventions could we apply that might, in a positive way, reshape the structure and function of that nervous system, building on a lot of this work in animal models that have shown us not only do we have the experience that we might all know that our experiences change how we feel, but also that they change the biology innately to us and how we can leverage that then to improve the lifespan.
Well, thank you. I think you guys both are doing really great and important work. And so I'm so glad that we get to talk about it a little bit more. Now that we've got kind of a groundwork for understanding the general goals and objectives, I would love to hear from each of you what you think is unique to your model or what it is about the animals or the humans that's allowing you to get at something unique in the research process that's especially important for understanding this topic. Why don't we begin with Millie?
Okay. Sure. So what I conceptualized sort of like this was a eureka moment when I was a postdoc is that most people that are interested in adversity are focused on mostly the majority of researchers actually focus on the offspring. But I view adversity exposure in the context of the mother-infant dyad. So we know that this is a bidirectional relationship. It's a bidirectional process. And I would argue that studying the maternal brain in the face of adversity is equally important, if not more important, perhaps. So in our research, we've identified that rat moms that are exposed to adversity have an increase in what we call harsh or adverse caregiving of pups. And this has been very reproducible. It's widely known. And we just kind of accepted as a field that this happened, but we didn't understand what was happening in the brain while this active adverse parenting was occurring. So my work as a postdoc characterized dopamine neuron firing activity in the ventral tegmental area, which is a huge, critically important area because it sends dopaminergic projections across multiple brain regions and governs processes like executive function, motivated behavior, and even cognition. One thing that animal models allow us to do is to use novel optical recording techniques so that we can measure neural activity in genetically defined subpopulations during maternal behavior. So this allows us to sort of have a better handle on what the maternal brain is doing during active maternal behavior, which is unprecedented. And that's where we're most excited to be going into.
Yeah, definitely. I mean, I think animal models in general give us a real great opportunity to capture information sort of while behavior is ongoing oftentimes, which can be difficult in clinical populations where we often rely on report and sort of after the fact understanding. So with that being said, let's now switch to humans. And what is it that the human model offers us on this topic that is unique and especially helpful when making advances in development treatments?
Yeah, so I'd like to tie the thread that you started weaving for us, Millie, where you're talking about, we know from clinical literature that adverse experiences, particularly during these really important times of our life, like maybe the maternal period, whether it's during the prenatal phase or the postnatal phase, they have this profound impact on our emotions, our health. And we can say that as humans, but then we don't necessarily know, okay, what is the underlying biology of this experience? And that's exactly what you're testing. And then on my end, I can take that information and kind of create a circle here where we can't ask your rodents how they're feeling, but we can then translate the paradigms you use into paradigms that we can test in humans. And for example, I use a fear-potentiated startle paradigm that was developed from a rodent model. And we could do things like expose the dams or the pups to stress and then look at differences in fear-potentiated startle. And we can do the same in humans as well, where we might ask them about previous experiences and then look at differences in startle responses. And we can close the thread by then saying, well, what is your experience like? And learning about how there are associations between what might be happening at the biological level and how someone is feeling. So then we can start targeting that through a treatment approach. And we might then even go back into the rodent model and test out whatever that new treatment that we think might work because it's targeting this circuit and seeing if that's correct, if we really are targeting that circuit with this intervention. And then does it produce a change in behavior? And does that translate from the rodent to the human's clinical experience?
Yeah.
Wow, what a fantastic, this is really great. And you're already sort of getting into my next question, which is I'd love to get even deeper into what the process of translational research really looks like. And you've sort of alluded to, reading about certain work, using that to inform your own work, and then hopefully that work that you develop then further influences additional work. And so I'd love to get your guys' opinion on the translational process generally and what you think the most effective form of translation is. We have this version you guys have just discussed about using the literature to inform your work and going back. That's one example. Are there other examples that you think are especially effective, like active collaborations with groups? Is that really important? And what about things like sharing data, allowing researchers from across different models access to data and attempting to integrate it? So I'd love to get your thoughts on that, and whoever would like to start can begin.
I guess I can go first because translation for me looks a little bit different, right, as a rodent researcher. So traditionally, people have thought as translation, including myself, as what can we learn from the clinical literature that we can try to capture in the rodents so that we can gain information that is not readily gained using human populations. So an example of this is the resource scarcity paradigm that my lab uses. We know that mothers that are living under high stress, low socioeconomic conditions have a higher incidence of these harsh maladaptive caregiving patterns. And we didn't know why that happened. So using the tools and the precision that we can have with these novel optical or circuit specific or cell specific tools, we can implement that. However, I do want to point out that rodent researchers have to be really careful because we never want to give the impression that we're mimicking a human condition, right, in the rodent. We do think that we can focus on little bits and pieces, constructs that are the most salient in the humans. And one thing that we can do is to start actually going beyond from meeting human researchers at conferences and continuing a conversation. So if anybody is an animal researcher, they should be identifying, where are the clinical findings telling us, like, where is this going? But also being proactive about asking, what questions would you like us to try to answer? Like, what seems to be, we know, for example, that there's many risk factors, mediating the effects of early life adversity. But we also know from meta-analyses and multiple human studies that there's certain ages that are more important and that there's certain types of adversity that are also more important. So having this bidirectional communication of how can we help, how can we unlock new knowledge that's going to help you unlock more new knowledge, right? And sort of keep this going. So I do think that having funding mechanisms that support and are explicit about supporting partnerships between human and animal researchers are very important, because I feel like in the current climate, it is hard to write a competitive, truly translational grant. And then finally, also placing this focus on translational research as something that is valuable, right? Like, and having that be recognized by university committees, scientific societies, and sort of creating that space and sending that message that we need better integration.
Mm-hmm. I completely agree with you about that point of better integrating us together instead of just reading each other's papers or talking to each other right now. We do need to actively work together to share data with one another, because like you said, the questions, one of our limitations is we can't test causality so easily. And maybe when we can, it will be a longitudinal study that takes 20 years to conduct for us to answer a question. And that could be a really important question that has implications for policy, that has implications for how individuals live their daily lives. So if we have an opportunity to have better clarity on the direction that we should be going in, and how do we make a worthwhile investment if it is going to end up taking us such a long time or so many people to learn from in order to derive that knowledge, being able to say, okay, in our human data, we found an association between adversity at this time period in life based on someone's retrospective recall and, it increased arousal or reactivity that might mean that that person is always feeling anxious or on edge and causing them to have clinically impairing symptoms. Well, then we can go back to the rodent model and to our colleagues and to say, okay, you have this ability to manipulate these different conditions. Can we see what is the directionality of this relationship? Because that has different implications for our treatment approaches. I also think having ecosystems, institutions, departments where the animal and human researchers are really close together is of benefit. And we actually have a facility like this at Wayne State University in Detroit, where I'm based, our iBio building, where we have wet lab spaces, we have clinical research spaces, and it's getting into this mindset, I think, that we're always working together, not, after we've published our papers and send them to each other, but throughout the whole research process.
I really am enjoying these thoughts, and I want to follow up a little bit on this, because you guys are both sort of alluding to we need better mechanisms, funding mechanisms, institutions to better support collaboration and allow this translational work to happen. And so I'm curious now, given the way the state of things as they are and the current infrastructure that we have set up, what would be your best advice for researchers who are looking to build collaborations, for how to pursue those avenues and to build really successful and fruitful collaborations?
I have a strong thought about this. I would say it starts with training. So we might be past the stage, but we're always lifelong learners. But maybe when our graduate students are coming into our labs, for example, or we're pretty early in our careers, but if maybe we're ever in leadership types of roles, steering the direction such that trainees are not just working in a rodent lab or the human lab, that they have an opportunity to rotate in other labs that are using cellular, animal, computational, human models, and they're seeing those approaches, even if it's just for a semester. And there are programs out there that encourage that rotational model. And I think even if we're not embedded in a program like that, we can encourage our students to take such an approach. Or if you're a new trainee out there, you can seek out these opportunities and talk to your mentorship team about it. And then, we can actively put in that work too, to go into each other's lab spaces, shadow each other's lab meetings. But I think it has to be taking this lifelong learning approach and getting that mindset from the very start and being deeply embedded in each other's research.
Do you have any additional thoughts?
Yeah, I love that idea. As an animal researcher, I get a lot of value from attending the ISDP, so the International Society for Developmental Psychobiology meeting. And as you said, we're a rodent lab, but we care about learning something that can help us improve the life of humans. And that is a value that drives our research. It drives our research questions. It drives our approaches. And animal research PIs can model that by bringing trainees and encouraging them to seek out these clinical researchers, so that this conversation can be started earlier, right? As you mentioned, during the training. I would also like to say that in our current climate, right, animal research is currently under attack. So one thing that if we want to be able to achieve this dream, right, of this better integration and this continued crosstalk between clinical researchers and animal researchers, we do need clinical researchers' support in voicing the importance of animal research for their discovery process, so that we can ensure that we are doing the best animal science that we can, and recognizing that it has a complementary value, not a competing value with human research.
Well, this is amazing because it goes right into my next question, which is how can clinical researchers better support animal research? As you said, it is a super critical time for animal research right now. And I think that most clinicians would agree that animal research is important, and most, researchers that study human populations would agree that they get a lot from animal research. But what are some things that those researchers who are not studying animal models can do to better support those animal researchers?
Write to your Congress people, write to your representative, but also promote. I know a lot of human researchers that take rodent findings and use it to design their own experiments, actually being more vocal about giving credit. This idea was found in an animal paradigm. And look, we see the same thing in the humans. There's value in just engaging in more direct scientific communication. And this doesn't have to just be limited to social media. I think it's unfortunate that a significantly large percentage of the American population can't tell you what a researcher does, or they can tell you that they don't know one. So just talking to your neighbor or your family members, having a 30-second pitch that you can give so that you can try to create this interest and sort of be able to reshape the narrative. Because if we don't engage in these scientific communication and these outreach efforts, then the public is just hearing whatever they're hearing from our current administration, right? And perhaps other organizations. So actually being an active driver of this conversation, I think is an easy starting point for anybody that does science.
And Lana, do you have any thoughts about this topic as well?
Yes, I think that we have to continue to have these conversations even close to home. Even we're here at the ACNP meeting, and we've surveyed our colleagues in the past and found that we do have colleagues even within our ecosystem that don't recognize the value and importance of animal research. So I think this just has to be a continuous conversation. And it really does start by acknowledging the foundational work of others. I think any scientist has had the point where they've thought that they've had a great novel idea, and they go back into the literature and find out that even if someone couldn't test it at the time, they had this idea 50 years ago. And I think it's true that with most of our clinical models, even if we're not aware of it, there's probably an animal model that exists in our historical record or is continuing to be used today that has informed our thinking. So I think even being self-educational and doing that work, even if you're beyond training phases, we're always lifelong learners. and sharing that with one another. I certainly also echo your call to contact policymakers, because the language surrounding animal research is something that's written into our budgets annually. There's language around animal research regulations in the Health and Human Services appropriations. So it's critical that, the language in that legislation is actually shaped by people who are doing the work and who are impacted by the work. The research that I do today with children and adolescents to help youth who have experienced trauma and adversity would not be possible if we did not have our fear potentiated startle model originally developed from rodent studies. And we continue to improve upon that model, which has profound implications for our gold standard treatments for PTSD and anxiety disorders as well. And we could not continue to advance if we left the rodent research behind and weren't able to continue updating those models, understanding the biology better.
So we started talking about this a little bit, and I want to get into it even a little bit more. I really echo everything you guys are saying about the importance of engaging and sort of getting in the room and speaking up and making it clear where animal research is important and how it's influenced ongoing research on both sides. And I want to sort of continue by sort of thinking again and expanding on this idea of how animal researchers and clinical researchers can better help the public understand the critical role. I think that for me, I'm of the opinion that part of the lack of support in our government and in our population is because there is a lack of understanding, and there are these other groups that are providing a lot of misinformation. And so what are ways and strategies that we can engage with the public in a more effective way and in a way that allows, maybe on an individual by individual basis, that to move in the right direction?
Yeah, I would say that great that people think that we have such computational abilities and clinical abilities that we can answer questions without animal models. I appreciate that maybe people coming from a completely pure perspective have such a high recognition of what our abilities might be. And the reality is that we are not at a stage where computational or cellular approaches could replace animal models. And there's just so much complexity in working with people with all of our individual variability that there is no way that we could function without our animal models. And this would actually have profound implications for treatment development and safety. It is so important that we're able to have those animal models in order for us to ensure that any treatment that we're going to ask somebody to invest in is safe and that it's effective. And there are so many people along the way who also ensure that the quality of life, the safety, the health of the animals who are involved in that research process is always monitored and always prioritized so that it can have high translational potential for human beings. So I think that I would first start by dispelling this notion that we have abilities that we just do not in order to answer really important questions related to health.
Yeah, I'd like to follow up on that and expand a little bit. So when we heard these communications, right, about this cutting down on animal research, et cetera, when you think about somebody like me who does developmental and maternal neurobiology work, my first instance was, some people were like, well, maybe you could just use cells or grow something in a petri dish or use organelles. It's like, how do you capture the changing of the brain, across development? Like, that is impossible to do with like a group of cells in a petri dish. Even the maternal behavior changes are driven by interactions between changes in brain activity and neural circuits governing all these behaviors that are important for motivated behavior. But it's an interplay between these neural circuit changes and hormones, which you can also not readily mimic, right, in a dish. So I do think that we need to do a better job at communicating what it is that we're gaining from animal models. And that is the first step. And that might look different for some people. For some people, it might look like going to high schools and giving workshops or giving talks. For others, it might be launching a science communication campaign or creating like their own labs, X or Blue Sky account. So there are many different ways, but we cannot afford as scientists to be sort of like set apart from the rest of the population. Like we do need to make these efforts to reach, these individuals that shape also and respond to these messages that are being given. And if we educate them, then we can have them become advocates for our research too.
I could not agree more with both of your points. I think these are really excellent thoughts and ideas that you're having. And I now want to sort of shift back to your own research a little bit. And I'd like to start with you, Millie, on this question, which is, what do you think are some of the misconceptions about animal research, even specifically with your own animal research, that you would like to, counter or talk about that are important for people to understand?
Right. So I think I touched upon this earlier, but we know that rats are not humans, right? So we are not trying to study postpartum depression in rats. We are trying to study a translational risk factor for postpartum depression, which is adversity exposure. So most people that I know that do animal work are very careful about this distinction, and we don't want that messaging to get mixed up. Another one is that we don't care about our animals, because if we did care about our animals, we wouldn't be putting them under conditions of adversity, or that we somehow value that animal's life less. So in animal research, if you don't take excellent care of your animals, which means minimizing distress, making sure that they're well fed, making sure that they're clean, you get noisy, unusable data. So it is in every animal researcher's best interest to treat those animals in the best, most humane way possible so that we can prevent waste, right? And collect data that is irreproducible, unreliable, noisy. So I think that those are sort of like the major 2 points. Yeah.
And now, Lana, I want to ask you a slightly different variation of the question, which is, what are some misconceptions or misunderstandings maybe that you've had about animal research in the past that through engaging with collaborations or translational research, you've thought differently about with some time?
Yes, I had no idea of the complex infrastructure that goes into taking care of these animals, ensuring that we reduce any of that noise that could contribute to potentially wasteful data, and really maximizing animal welfare. This is messaging that is really countered actually in the public, and the messaging that we see in the public is just not accurate. There are veterinarians, there are husbandry staff, there are the researchers themselves, there are administrative staff who oversee ethics and safety. There are also building maintenance staff who make sure that that whole larger environment is clean and safe. And there are many, many more people who put so much time and energy into the care of those animals. And these are all people who have a great passion for animals in many cases as well. I also did not know about some of the retirement programs for our animals that are in research, which I thought was the coolest thing ever, that there are many animals that might go to sanctuaries after they have contributed information, data to science, that they are housed in for the remainder of their lifespan in the best quality conditions, and that there are even adoption programs, maybe for rodents or other animals that have been in research to go on and have another life with a new human friend after all of these other people who have loved and cared for them so much. So I just thought it was really cool to learn more about the care and the people that really make all of this work possible, aside from the fantastic researchers who are the ones that we might most closely think of as interacting with the animals. But there are just so many other people who really put so much into making that work possible.
Well, I really want to thank you both for such a phenomenal conversation. And before we wrap and close for today, I just want to open the floor. And if there's anything else that you wanted to talk about or make a point about as it relates to animal research, human research, the translational process in general, I just want to give you the opportunity to share anything else additional that you would like to. No pressure.
I'd say gratitude.
Yeah.
Gratitude to our fellow colleagues, gratitude to our animals. The work I know also benefits animal lives as well, not only domesticated animals, but also even wildlife. So I think I'd want people to know that the implications go far beyond, the work that we do in adversity and thinking about mental health outcomes for people across their lifespan. Yeah.
Well, I have so appreciated hearing both your perspectives today on animal research, the translational process, also about how these research processes help us better understand adversity in early life and risk or resilience for psychopathology later in life. So I just want to give a really wholehearted thank you again to Dr. Rincon-Cortes and Dr. Ruvolo Grasser for your time, your openness, and your wonderful insights on this topic. And of course, thank you to everybody who is watching or listening as we learn about how these parallel research avenues take us from model to medicine. So be sure to tune into our future episodes as we continue these important translational conversations. And until next time, remember to stay curious, intentional, and thoughtful in all your pursuits, but especially in research. This Lab Rat Chat special series, Translational Conversations, is made possible through support from Biomedical Research Awareness Day, a program of Americans for Medical Progress, and the American College of Neuropsychopharmacology.